Hi all, welcome back to Microbiology Lectures. Today we're going to discuss really the structures of bacteria, the cell wall structure, and a little bit about kind of the methods that we use, at least one method that we use, to observe them. Okay, so let's jump into it and start with the history of microbiology, or a historical individual in microbiology, and that's Hans Christian Joachim Graham, who was a physician that worked in morgues in Berlin and was tasked with determining what's the causative agent of pneumonia. So people are dying of pneumonia, being sent to the morgue, and he's collecting tissue from the lung, or at least mucus from different regions, to see if he can figure out what's the causative agent of pneumonia. And so this is a picture of Hans Graham. And so while he's doing this, he realized, as many scientists and physicians and individuals during the 1800s and the early 1900s realized, that a lot of these microorganisms that they're dealing with, you can't see them. They're clear. And so you have to devise some way to stain the material, the outside cell wall of these organisms, in order to get a look at them and determine if they're the same species, or at least same type, that you find in every single individual that may have died from pneumonia. And that's exactly what Graham did. Graham devised the method that we now call the Graham staining method, and that was, you take these bacteria or microorganisms that are in a sample. You apply crystal violet to the sample. Crystal violet will dye peptoglycan, and so then after that you add iodine. Iodine will typically kind of open up like fats and help with binding up excess crystal violet. And then you can clear that excess crystal violet, the stuff that hasn't been absorbed with peptoglycan. You can clear that by using ethyl alcohol, right? Which means that the bacteria that don't have a thick cell wall of peptoglycan, would retain just a little bit of crystal violet, but not enough to be like all purple, I guess, or all dark blue. And then you can apply a stain like saffron, which dyes lipid-based structures. It'll dye it pink or light red, and then you can determine the difference between two different types of bacteria: a gram-positive, which would have a large, thick peptoglycan layer, and a gram-negative, which has a very small peptoglycan layer and then a capsule on the outside of that. And so by devising this, we now know that there are really two main different kinds of bacteria based on this staining method: those with thick peptoglycan layers in their cell wall, which we call gram-positive, and those with a thin peptoglycan layer in their cell wall and a capsule that covers that, which we call gram-negative. And so today, in many labs and pretty much everywhere in the world, Gram stain, this process is used to stain microorganisms to see if we're talking about a gram-positive or a gram-negative group of bacteria. And the reason why that's important is a lot of our antibiotics are geared towards, or work based on, what is the cell wall structure. Okay, and we'll come back and talk about how structure determines function in this case. Okay, so if we were to, go through the process and do a gram stain in an unknown bacteria sample, and we get something like this. Like, for example, you might have Staphylococci or Staphylococcus groups of cocci bacteria, that are clumped together. So we call them Staphylo cocci or Staphylococcus, okay, because they're circular, clumped together. Okay, we could also tag another term onto that. We could say they're Staphylococcus gram-positive bacteria, okay, which then tells us what their cell wall is made of. Okay. On the other note, you can see there's another type of bacteria here within, this one slide. These guys are not circular. These guys are rod-shaped, and so you would call them bacillus or bacillium. And so then you could say that, they look to be singular. They're individual. They're not chained together or anything like that. So you could just call them, a bacillium or bacillus gram-negative bacteria. And so that allows us to categorize two separate types of bacteria. And then, you could go from there to try to figure out which one might be causing the disease, or which one might be native, and whatnot. And if you've looked at enough samples, you would know which is native and which is a foreign body in that individual, and causing disease agent for that individual. Okay, so let's look at prokaryote versus eukaryotic cells. Again, we talked about this before. Prokaryotic meaning before nucleus, eukaryotic meaning true nuclei, or at least that's a rough definition of both. Okay, so prokaryotic, we're talking about bacteria and archaea. Eukaryotic, we're talking about animals, plants, protozoa, and fungi. Okay, and yes, algae sometimes is broken off because there is algae that are plants, and there are algae that are protozoa, and then there are algae that are sometimes linked to fungi in their own kind of special group called lichen, and sometimes we throw lichen underneath fungi, which is like a fungus and an algae together. So anyways, classification sometimes it's a little bit difficult, but nevertheless, all these organisms have a true nucleus. Okay, so if we look at the differences between the two groups really quickly, you're going to see both groups have genetic material, typically in the form of DNA. One group lacks a nucleus, the other one has a nucleus. Okay, so in bacteria and archaea, we call this region where you'd find the chromosome the nucleoid region, but there's no membrane around it. Okay. You can also see in the eukaryotic cell, you can see a bunch of organelles. So here are some mitochondria. You can see a Golgi complex or Golgi apparatus, the rough ER or the rough endoplasmic reticulum there, lysosomes. Okay, and then we jump over to prokaryotic cells, and you see, they have ribosomes also. But remember, ribosomes are not membrane-bound organelles. Ribosomes might function like a membrane-bound organelle, but they're not classified as a membrane-bound organelle, or for that matter, really an organelle. Their function is to translate proteins, so translate genetic code into amino acid chains, which eventually become folded up proteins. And then you can see also in the prokaryotic cell, they're going to have a cell membrane, then a cell wall, and then some of them have a capsule, and those would be the ones that stain gram-negative. Okay. So there are similarities and differences between the two, and that really has a lot of consequences in designing and utilization of antibiotics. If we have an antibiotic that destroys cell membranes. Okay? And we're using it to destroy prokaryotic cells, bacteria that might be causing disease or something like that, then what's to say it's not going to destroy the eukaryotic cell membrane also? And so if that's the case, it would destroy both, and then the eukaryotic individual is harmed by it. Okay, so it definitely, the figuring out the differences and the similarities between the two definitely drive modern medicine. So the idea is to choose things that prokaryotic cells have, but eukaryotic cells don't. So like maybe a capsule, potentially a cell wall that's made out of peptoglycan, okay? A certain type of ribosome. What's not pictured here is like ATP synthase, an enzyme that is needed for the organism to generate energy. If that enzyme is different enough from a eukaryotic enzyme, then maybe we can target that enzyme, and we'll come back and talk about that. That's kind of a modern, that's like a new way that we're trying to generate antibiotics, is to attack ATP synthase. Okay, so prokaryotic cell structure, just general. I'm just going to do a general overview, and then we'll dive deep into these in a future lecture. But just in general, the prokaryotic cell is kind of made up of, often is made up of kind of 3 layers. So you have a cytoplasm, and this is where ribosomes and the nucleoid is going to sit and kind of float around in this cytoplasm, which is kind of like a wet jelly like substance, lots of water, but there's also proteins and sugars and salts and all kinds of things that occupy the cytoplasm. Okay, you have a single chromosome, single circular chromosome, and the nucleoid region is just the region that you would look to find it. Okay, so you can see over here in this electron microscope picture, you can see this light region is that nucleoid region where you would find the genetic material. Cytoplasmic membrane is the membrane that basically protects the cytoplasm, or is directly connected with the cytoplasm. So this is the first layer that you would see going out from the cytoplasm. And that cytoplasmic membrane, we'll come back to it and we'll talk about the similarities between the different groups later. And then all bacteria and archaea have a cell wall. Now what it's made out of differs between the groups and even within groups, but they have that cell protective wall. And I just want to make sure that we call in plants, they also have a cell wall, but it's different than the cell wall of bacteria and archaea. Okay? In fungi, they also have a cell wall. It is different than plants, bacteria, and archaea. Okay? So just the cell wall structure, the name is just this protective layer that's outside of the cytoplasmic membrane, or outside the membrane. And then some bacteria will have a capsule, that even another layer on the outside, and we'll come back and we'll talk about this. Some of them will have a way to move, like a flagellum or a flagella. They might have multiple flagella throughout their body, or they might just have polar flagella, and maybe they have a few on one end. Okay, so there's lots of different setups. And then here you can see these other structures, which are kind of unique structures that a lot of bacteria have, and that is a pilus, or the pili. And these go all the way through. In this case, you can see that this one has a capsule, so it goes all the way through the capsule, the cell wall, and the cytoplasmic membrane, so it can encounter the cytoplasm. where you might find plasmids and other things, and it has the capability of exchanging plasmids, small chunks of DNA, between individuals. So it's a way of bacteria, some species of bacteria, to basically have sex without creating offspring, but changing their genetic makeup. And so one bacteria can pass a plasmid to another bacteria, and maybe multiple plasmids are passed back and forth, but it just increases the genetic diversity of the bacteria. And it's been known that in some cases you can even get cross-species passing of plasmids, so from one species to another species, and that makes it very interesting. The other piece that makes it interesting is the genetic material that's being passed, the plasmid. We've looked at a lot of the genes on the plasmid, and a lot of those genes control for things that help with surviving extreme environments like temperature, salinity, help with the bacteria's immune function, or at least defenses. So bacteria, we'll get back to this, they have what we call efflux proteins, which are proteins that expel waste products. And so a lot of plasmids control for the proteins, the efflux proteins. They have the genetic code for those efflux proteins. And so some of the antibiotic resistance that we see is when an antibiotic gets into the bacteria through the cytoplasmic membrane into the cytoplasm, that these proteins pick it up as foreign material and spit it back out. Okay, so the antibiotic actually has no way of destroying, like if it's an antibiotic that prevents the chromosome from replicating, then you might have that antibiotics being spit out before it can prevent that chromosome from replicating, or prevent the bacteria itself from replicating. Okay, so there's lots and lots of things. So cytoplasm I already talked about. Nucleoid region, that's where you find the chromosome. Locomotive appendages, not all of them have them, but some of them have them in different regions, so they can be, like I said, they can be polar, they can be on one end, they can be placed in the center, and we'll talk about what we call when we place flagella in different spots. There's special names for them. And then I just talked about the pilus, or the pili, and their role with plasmids. So in a nutshell, that's kind of what we talk about when we talk about prokaryotic cell structure, big picture. Okay. Next, we're going to dive in to what is the cell membrane, what is the cell wall made out of, what is a capsule made out of, and what role might they play in the organism's life. Next time.